Welcome everyone to today's webinar sponsored by Collaborative Drug Discovery, provider of CD Vault and BioHarmony and BioHarmony Annotator. I'm Frank Cole with C2D. This afternoon we have a great cutting edge discussion with our panelists as you see them here on the screen in their webcams. We're joined by Dr. John Griffin, Dr. Jeff Warrington for a discussion on modern machine learning methods applied to drug discovery data, moderated by John Overington. I trust that you all have had a chance to read up on our panelists and their bios as you were registering for today's event. Welcome, panel. I would love to also ask all of you who are in attendance to pose a question in the Q&A panel so that we can save those questions to the end for panelists to address. We will save time before the top of the hour. At this time, I'd like to turn it over to John Overington, who will lead and moderate today's webinar. John, all yours.
Thank you very much, Frank. So hi, John and Jeff. It's a real pleasure being here today. Let me get right down to it. I guess one of the big questions for a lot of people is exactly what is machine learning and AI as applied to drug discovery. So maybe definitions from both of you. Jeff.
Great. Hi. Good morning, everybody. My name is Jeff Warrington, senior scientist at Atomwise, and we are one of the leaders in using machine learning and AI for drug discovery. I like to think of machine learning as a broader category of any technique that uses drug discovery data in a non-parametric way to predict upon experimental results. If you go to my next slide. By way of introduction, Atomwise was the first company to use convolutional neural networks for drug discovery. To date, we've raised over $170 million to prospectively use AI algorithms in real drug discovery projects. With projects with the top 10 pharma companies and academic and hundreds of academic collaborations I will talk a little bit later about, we have over 750 drug discovery projects in flight. We've seen real successes in our academics projects or AIMS projects with over 75% of our collaborators reporting hits from initial screens. So in terms of your question, if you go to the next slide. At Atomwise, we focus on structure-based drug design using AI algorithms. I want to compare and contrast that to traditional, for example, docking methods. That use a lot of complex equations to estimate binding affinity of small molecules to targets of interest. These equations are highly complex and have a lot of assumptions built in with statistically derived terms added in. At the end of the day, the equations do rely on a lot of human intuition to ascertain the most relevant physical interactions. So this is a compare and contrast to traditional statistical methods. Next slide, please. So our platform technology, which is a three-dimensional convolutional neural network for drug discovery, uses a similar technology to image recognition. So instead of voxels for input, instead of pixels for input in image recognition, we use voxels. And instead of facial features such as eyebrows or chins, we recognize either atoms or empty space. So that's briefly our platform technology. If you go to the next slide, please. So is deep learning for drug discovery. There are several advantages to using machine learning models in the drug discovery space. For one, the models generally have best-in-class performance when compared with other classical approaches implemented. Generally, deep convolutional neural networks are great in finding generalizable, predictable patterns. And I think from the graph, you can see that deep learning methods increase logarithmically, the performance of deep learning methods, I should say, increase logarithmically based on the volume of the training data. So the more data that you have, the more the deep convolutional neural networks can learn about the problems you're trying to solve. I think that provides a brief overview and happy to entertain any questions later. Over to you, John. Thank you.
Thanks, Jeff, and thanks, John, and thanks everyone for attending. I appreciate the chance to participate. Good morning, good afternoon, good evening, depending on where you're joining from. So I think of artificial intelligence as functions or algorithms that are derived from the application of machine learning to training data. And so these mathematical or algorithmic models may then be applied to problems, to address problems of interest. So for example, in the context of drug discovery, AI allows us to make predictions of molecular activities, biological activities, and properties at speed, scope, and scale that are difficult and practical or impossible to achieve in the laboratory. By way of introduction to Integral Health is leveraging integrated biology and advanced technologies to engineer and deliver better medicines to people. We are presently operating in stealth mode, but it is fair to say that Integral is a patient-centric company with the simple yet ambitious mission of delivering medicines that matter to patients and increasing the scale and scope of health impact. If we go to the next slide, please. We are an integrated team of experienced drug hunters, drug developers, data scientists, and operational executives, leveraging AI to discover new medicines. As shown here in this figure, data lies at the center of essentially everything we do, as it provides the forum around which people with very different backgrounds and skill sets communicate and collaborate, as well as a field with which to test and refine algorithmic, computational, and experimental approaches. And that's my introduction.
Thanks very much. So again, for both of you, what would you say the scope and limitations of AI methodologies are? What do you find most useful today? How is it different to the past, and how, looking forward, do you think it'll get better? So Jeff?
All right, thank you. Yeah, I think if you advance to the next slide. Believe.
Yes, I apologize. I missed some text here. If I may apologize for this, if I may go back and complete some introductory statements.
Absolutely. Sorry.
Yeah, so thanks, Jeff. Thanks, John. So I joined Integral Health from a company called Numrit, with which some of you may be familiar. And at Numrit, I worked with colleagues with expertise in machine learning and computational expertise, over a period of 15 plus years. And at Numrit, we collaborated to build and apply a platform based on advances in ligand-based predictive modeling. And so as a complement to Jeff's introduction to structure-based methods of various kinds, ligand-based modeling is where training data for molecules that have properties of interest or not of interest are used to curate, from which we curate training sets. We can then take these training sets and apply different methods of ways to represent that information in a way that a computer can understand. And to these representations of the training data, we can then apply machine learning algorithms properly matched to the representations to derive these statistical models, these AIs that can then be applied to a number of problems of interest in drug discovery. The advantage of ligand-based methods is that they are quite general. In fact, that they can be applied to many phenomena of interest: activity, selectivity, ADME properties, toxicity. One doesn't require having the structure or structural information of the target. In fact, you don't even need to know what the target is, because this approach, of course, may be applied to phenotypic data. And so that's a great generality, because it really does allow you to span questions ranging from target discovery all the way through lead optimization and, say, in small molecule drug discovery. But of course, it's not without its challenges. To properly represent molecules in ways that are important for supramolecular interaction with their protein or DNA targets or other targets requires one to be willing and able to deal with very complex representations that capture the way small molecules behave in time in 3 dimensions. The other challenge here is, of course, the data, and we'll talk a lot about data today. And the fact of the matter is, the data that we have, as Jeff mentioned, from the medicinal chemistry and medicinal scientists is small data, right? It's hardly ever big data. It's probably never going to be big data. In addition, that data is typically derived from many different sources. And even from single sources, the data is very noisy because it pertains to biology, and is also very biased because it is populated by molecules that chemists decided to make, were able to make, had a chance to test, and decided to release data about. Right, and so those are challenges that we had undertaken and addressed at Numrit. And I think the bottom line for us is that depending on the problem at hand, we try to match those to the specific question, the specific the specific predictions we were trying to make. So the representation algorithm pairs apply to different things that are relevant to drug discovery, including deep neural networks, but also including many other kinds of algorithmic and mathematical approaches. So thanks, sorry for the interruption.
Thanks very much. And just to refresh all of us of the next question, maybe both of you could say what the scope and limitations of AI methodologies are today. What do you find most useful? What's different to the past? And then looking forward, where do you think the roadmap is for things getting better in the future? So Jeff.
Great, thank you. So next slide, please. So I think unlike many other domains where there are vast seas of data, you can think of recent advances in self-driving cars, or you can think of game data such as chess or Go, where you can infinitely train on self-constructed games. Drug discovery data is relatively sparse and very expensive to acquire. In addition, today, we believe through enumeration techniques, there are 10 to the 60 possible drug-like molecules that are synthesizable in chemical space based on a very limited number of atoms. So in fact, there are probably even more than that. We only know about how about a few millions of these potential or small molecules and how they interface with biology. I think what's different from the past is that now there are billions of unique and orderable compounds available, which completely overshadows even the largest pharma libraries or repurposing physical screening libraries that there are today. So methodologies such as machine learning, deep convolutional neural nets that can generalize well into the unknown will be very powerful in finding new leads for drug discovery. Next slide, please. As I mentioned earlier, prospective testing is the key. For this goal, we've really set up a global initiative to collaborate with leading researchers in both academia and industry through our AIMS program. And we're providing access to the technology to academic researchers worldwide. Through this program, we work with researchers to design a virtual screen of millions of commercially available compounds and then procure them. With many of these collaborations reporting back now, we're happy to report an overall 75% success rate in terms of finding small molecules that our collaborators qualify as worth pursuing. Next slide, please. I think there are real unmet medical needs in terms of drugging the undrugged. To date, there are only 4% of the estimated human genome that has drugs for it. But there are 4 times that number that have some disease evidence, and even the remaining 80% are quite simply just unknown. So I think today, coming back to your question of what's different today is there's a tremendous opportunity for AI algorithms to be developed and find leads for even the most challenging targets. Next slide, please. So today we're operating our platform technologies not only in target hit, but also in hit to lead and lead optimization, where we have to refine models for these tasks. Will undoubtedly lead to getting to better medicines to patients faster. And I think a key area what's different today is in terms of ADME, absorption, distribution, metabolism, and excretion data is extremely sparse, as John had mentioned earlier. So we continue to develop algorithms that are better suited for these low data situations. I think a nice analogy is you can think of a child learning about a cat. You really don't have to show the child every cat on the planet before they recognize what it is. Only a handful of examples should suffice. So I think new developments in these low data regimes will be extremely useful as we advance programs to the later stages where there simply is not a lot of data. Thank you. Over to you, John.
Yeah, thanks, Jeff. I can only concur that AI provides new tools, new capabilities to allow us to attack these difficult problems in ways that have not been possible before, as well as to greatly speed the scale, to speed up and to operate at scale against them, against the properties that are more easily addressed. So I would just like to add that I have the feeling that it's as much who is different in our field now as it is what is different. And so, working in teams of physicians and drug hunters with data scientists in very close collaboration, these people, we have typically very different skills and interests. For doctors, it's diagnosis. For data scientists, it's prediction. And for drug hunters, it's explanation and directionality, respectively. So this is very exciting and powerful from the perspective of designing, discovering, and developing drugs. But it does come, I have to say, with some interesting cross-disciplinary challenges. So, for example, I have been in meetings where the topic of discussion turned to APIs. And I have to say that half of the participants in that meeting were convinced we were discussing active pharmaceutical ingredients, of course, right? And the other half were just as convinced that we were discussing application programming interfaces. So integrating, finding common lexicography and integrating the insights of such a range of contributors using data as a kitchen table around which we sit and argue into AI platforms to ultimately serve patients represents really a key frontier for development in our field.
John, Frank, again, I'm going to interrupt just for a second. We're going to take a short pause to poll the audience. We have two polls today. So Charlie, you want to launch that poll?
I will do. All right, attendees. So kind of to help our panelists gauge the audience here, we have a large number of attendees coming in. We'd like to ask you, are you currently using or applying advanced AI techniques in your research? And as a two-part question, are you achieving results? So please make your selections here. Either yes, you are using these and achieving great results, or yes, you're using the techniques of AI, but the results are still being determined. Of course, the third option is no. So we'll let the attendees take a moment to complete their selections. They're still very busy finding the buttons and voting here. Appreciate your participation here. All right, everyone, I'm going to end the polling in 3, 2, 1. And here I will share the results with everyone. And panelists, you can see 59% of the attendees responding are not using the AI techniques. 26% right there in the middle of yes, they're perhaps using techniques of artificial intelligence, but the results are still being determined. So I don't know if there's any insights that you can draw from this, but good information to know, at least as we continue through the rest of the webinar.
Thank you very much. And now back to the questions. Maybe both of you could describe what you do at your current organization and how this has changed to where you worked previously.
Great. Thank you.
Over to you first, Jeff.
Great, thank you. So with my current role at Atomwise, I'm really happy I get to use the cutting edge of machine learning and AI in drug discovery, which is very exciting, but it's an evolving technology. We have an expression which is we build the plane while we're flying it. So, I mean, there's a real challenge in that we are constantly seeking to adapt and evolve the technology, but also we have pressing unmet medical needs. So we need to also apply this evolving technology in real time. And it's exciting and it comes with its own challenges. So that's my current role at Atomwise. Prior to joining Atomwise, I was at Cytokinetics, which is a biotechnology located in South San Francisco. They take a different aspect in that they are uncovering new pathways in human biology and seeking to develop new therapies from those pathways. The focus there was less on developing new in silico technologies, but more in developing biotechnology using available tools in terms of small molecule drug discovery. However, we were very successful and put a number of novel therapies into the clinic using that approach as well. So two very different approaches, I guess, to the problems in drug discovery, and both with their each unique challenges. Thank you. Over to you, John.
Yeah, thanks. And certainly at Integral, there's really strong philosophical alignment between Integral and Numerate, so which was really a key driver in our decision to join forces. And so in the context of our therapeutic discovery programs, we really have implemented, full scale of in silico predictions that really represent in silico assays that we can use to complement or in many cases replace laboratory experimentation to increase the speed, the scale, the scope, and the confidence of success in our programs, and which of course is highly enabling. And the other thing I'd like to say is what's important is that one finds a way to harmonize and resonate your experimental efforts with your computational efforts, right? A computational, you can evaluate billions of molecules in a relatively short period of time, given the scale of compute and the efficiency of algorithms that are available. Okay, but if you're only able to, but that really, you need to verify those results ultimately, in the laboratory. And so you need to have some level of resonance between the scales. They can be greatly different orders of magnitude, but it can't be, in my opinion, 1 billion down to 2 or 1 billion down to 3. You do need to be able to have confidence that you can test and verify the properties that you have. But in the context of, say, very precious low throughput assays, you want to be able to apply all of the tools at your disposal in order to select molecules to put into such precious assays. Going back previously, my previous company, Theravance, set out to and succeeded to exploit principles of multivalency in small molecule drug design. And this was really leveraging insights that came from my former Stanford laboratory and the laboratory of George Whitesides at Harvard. So the multivalency provides enhancements in target affinity and selectivity, which Theravance pursued, with more traditional medicinal chemistry approaches, with very little input from advanced computation, but as Jeff said, also very successfully. I mean, Theravance has discovered, a couple dozen drug candidates over the course of its existence, which is a pretty high rate of success as well.
Thank you very much. I'm just skimming through some of the questions coming in. This next one sounds really relevant for this. So both of you again, when does deep learning work better? Jeff first on this.
Great. Yeah, so I think, it sounds a little bit obvious, but I think when there's more data, but I think what's really, when deep learning really works well is when there's vast fees of data. But I think when it really works well is especially when there's negative data. Regimes where machine learning works well is that digesting complex data where simple models just don't work. And we need a more non-parametric understanding of the underlying data architectures. We have to be careful with overfitting in those situations, of course, and very careful with regularization and understanding what the machines are uncovering in terms of the relations to data. And more so, we have to understand how to interpret the results carefully so that they're learning about the right things. So in answering your question, I think it works better, and especially in places where there's a lot of negative data and not just positive cases.
Yes, and I concur. So negative data is very important, and it certainly has different and greater importance, as in my opinion, as you move further down in your discovery programs through lead optimization, where you're looking at small changes relatively to just general screening. And the importance of negative data, speaks to the fact that really today's machine learning approaches are really all about discriminative modeling. Okay, not a consensus model where we're trying, to overlay molecules to understand what is common among a set of active compounds. It's really what differentiates an active compound from an inactive compound, or a more active compound from a less active compound. And that's why, to Jeff's point, the negative data becomes very important. The other thing to say about deep learning is what's in other unsupervised methods is where you don't have a good idea of what good representations might be for the phenomenon you're trying to model, or you realize that there is bias in the representations that we are using based on our knowledge as medicinal scientists or chemists, etc., that actually don't allow us to take full advantage of the subtleties that are present in that data. So using a more unsupervised and unbiased approaches, it provides an advantage. But overall, and I tried to reflect this in the slide I showed earlier, is that it is important to approach things pragmatically, to select and develop optimized pairs of representations and algorithms in light of the data that you have and the problem you're trying to solve. Another thing that I think it's worth remembering or realizing is kind of one of the primary assumptions of applications of machine learning and machine learning algorithms is that it's assumed typically that test sets are drawn from the same distributions as the training sets. So in other words, that the resulting model is really meant to interpolate with the domain of applicability defined by the data rather than to extrapolate from it. Okay, so this poses a challenge for those of us who are tasked with inventing things that really supersede the prior out and fall outside it. And it's clever application of, again, representation algorithm pairs and use of those to actually have techniques where, in fact, you believe you can extend outside the domain of applicability as strictly defined by the training data that you have.
Thanks very much.
John, before you go on, let me just remind people, I'll go back a slide here, that this is the place to remind yourselves to pose your question. We've got a lot of people submitting questions, which is awesome. So go to the taskbar and find the Q&A icon and click on that, and that's the best place to get your question logged in. So back to you guys.
Great, thanks very much. That was a great entree to the area of data by both of you. But how do you ensure data quality is enough for machine learning model building and development, testing, and so forth? So Jeff.
Great, thank you. As I'm sure you can attest to, and you have a lot of work in this field, data quality is of utmost importance in model building. Some really get drawn into the latest algorithm or the newest approach to solving problems that are unique. But I think the most important piece in all of this is the data quality. For us, we really like to have data represented from multiple sources and being corroborated, which unfortunately is often not the case and very difficult to do in drug discovery. Not a lot of resources are spent to repeating things, to the nth time. There's a lot of manual labor, and at Atomwise we have a team dedicated, a whole team, informatics team dedicated to looking at the data and making sure it's consistent with things. Abe, our CEO, points out to a number of cases where, for example, nanomolar and micromolar or nanomolar and millimolar are just a simple typos, but in terms of numeric means, that's very, very large differences in terms of what the actual amount is. So I'm really lucky in terms of, I find my role is more on the application side. So I'm a consumer from the cleaned data products from my coworkers, and they present us with algorithms that are really trained on clean data, and it's really of the utmost importance to ensure that what's going into the algorithms is well represented. Over to you, John.
Yeah, I agree. Data is so paramount, and I think Jeff and I really do benefit from contributions that CDD CEO Barry Bunin and our distinguished moderator John Overington have made in this field, and then they have made them over a period of decades to the fields of data and data science. So Barry's former company Libraria, I believe, seeded an entire industry where the product is systematically curated medicinal chemistry data, comprehensive data. And John, I think, really is the primary driver of making such data openly available when he was served as the Campbell Service Team's leader. So, these data are really key enablers for democratizing discovery through application of data science and artificial intelligence. It's really not possible to overstate the importance of having access to this kind of data. As to the data itself, I agree. One must begin with the assumption that some of it will be wrong, in addition to being noisy and biased, as I mentioned earlier. So we also have teams of data scientists who really focus on the data, understanding its importance, and we've developed and we apply all sorts of automated data processing workflows that really flag many of the potential issues in data for review, ranging from the signs to the typos Jeff was discussing. Many stereochemical depictions in the data are wrong, some things that we've seen over and over again. But you know, sometimes hand curation is required, or at least, you know, chemists need to look at the data to quality control it. But the automated workflows can point out just where they should be looking and where it's reasonable for them to spend their time QCing that data. But it's also possible to, you know, to think about and understanding the level of noise in your data, and so that you can apply appropriate pairs of representations and algorithms that have intrinsic robustness to that noise. And that is something that helps you, even in cases because you're not going to identify all the problems. You need to accept that and work in that regime.
Great. We're going to sneak in one more poll before John continues. Charlie.
All right, everyone. I will launch a new poll. It's a quick one. What would help you most to leverage advanced AI techniques in your research? Are you thinking you would need more access to more data, access to collaborations, or access to actual algorithms? What would be most beneficial for the attendees here? Lots of responses coming in. I can feel everyone sitting on the edge of their seats to see the results of this one. All right, seems to be slowing a bit, so I will now end the polling in 3, 2, 1, and share the results here. So access to more data seems to be the top choice at 41%, but 37% access to collaborations, of course, very close there. Access to algorithms didn't fall too far behind. So I don't know if that's surprising to the panelists. Any thoughts on this spread of what attendees feel they might need to leverage AI techniques in their own discovery?
Yeah, no, I mean, I think access to more data is certainly very beneficial. And I think that really goes well into the next question. Unless John has other
so I think a
comments.
great lead in there.
Yeah.
Maybe both of you can outline what the importance of FAIR data is. Now, I've just noticed, I guess, that FAIR has got AI right at the center, so maybe there's a message in there. Yeah?
Yeah, I mean, I think it's really important to balance the proprietary nature of data, and as I had mentioned, especially drug discovery data, which is very, very expensive to acquire. So there's not a lot of incentive to just share that for free to the world. But you know, FAIR data being findable, accessible, interoperable, and reusable, it's important to the fields that FAIR data be accessible to the world for development of new algorithms. And it's really not obvious in a lot of models in the drug discovery space that you can reproduce the same results with the same data, actually, especially if they're trained on different subsets or looked at in different ways, or represented, as John was getting to earlier. If the molecular representation is different with different models, you can get very different answers. In a lot of computational work, I find, especially in literature and coming out of big pharma companies, where they produce papers about new methodologies that involve a large amount of proprietary data. I think while these are of benefit to the organization internally and to the authors, of course, in terms of getting to publish a paper and present at conferences, the new algorithms that actually use a lot of these proprietary data are really unusable by everyone else. And I think whenever there is an opportunity to make data and algorithms available for recreation and use by the public, I think it will definitely benefit the field.
Thank you. And now over to you, John.
Yeah, I would agree. I mean, really, FAIR data is the critical third leg, along with compute and algorithms that, you know, support progress in our field. It enables the entire community of investigators and provides, again, the central element around which we can collaborate, you know, and around which we can compare results, and we can also reproduce results. Again, in order basically to do science, we do need to have common data sources and robust data sources. Horizontally extensible compute is readily available from Amazon, from Google and other providers. And many of the advanced machine learning algorithms, deep learning algorithm cadres, right, those are also publicly available. Really, it's the data that is the key piece that is probably the most important. And the more that becomes available and remains available, the more progress it will support.
Thank you. So we've heard, I guess, in a bit of focus on your particular group's research and science, but looking more broadly, what would you say are the sort of 2 or 3 most interesting applications of machine learning in drug discovery today? So John first for that one.
Yeah, I am happy to share several things that are shown on the next slide. It is a prompt for this, but really I want to emphasize that these just represent a fraction of the activity and the progress that's being made in our field. It's really breathtaking, I think, at this point. In the upper left-hand quadrant, going back to something I mentioned earlier about technology developed at NUMRIT, this is an issued patent for ranking-based learning, which is one of the approaches that Nigel Duffy and our colleagues at NUMRIT developed to address noise and bias in training data from biological systems and biological assays. And this is, it turns out, it's a very good way to really remove the underlying sources or conundra that are associated with the bias and the noise in the data. And really, it comes from the perspective of saying, well, what is really the question the medicinal chemist is really asking, which is, what molecule should I be making next? Well, that's really a ranking-based question as much as it is a quantitative number-based question. The second in the upper right-hand quadrant is the realization and the various demonstrations now that biological data, and in particular phenotypic data, may serve a variety of useful purposes when you use it in the context of other data, such as on target and biochemical screening, for example. So there's this paper from Hugo Kellermans, Jensen, and his collaborators that speak to this. And it's relatively recent. It shows, we've done all this high content screening. In what ways may we use it for other purposes? And maybe some of you maybe understand, right, that company based in Salt Lake City, Utah, Recursion, has set up a data factory to produce such types of high content data to use for many, many different purposes in drug discovery. It's really interesting. In the lower left-hand quarter is something I find, it's rare that I believe is, among the places the field is going very rapidly, which is the purpose-driven integration of systems built on different kinds and levels of data and algorithms. And so in this report, then Nelson, Butte, and Baronzini showed that you could link systems pharmacology network, which they call SPOKE, with electronic health records for individual patients, okay, using unsupervised learning. Okay, so now we're not connecting the dots, we're connecting patients and systems biology and use back integrate that you can imagine to drug discovery at some point. And finally, in the lower right-hand corner is just the homepage for the Alliance for Artificial Intelligence in Healthcare, AAIH, which is a global advocacy organization founded by my colleague Brandon Allgood and other technical and business leaders in our field. And I think the very existence of this organization now speaks to the explosive growth and the growing influence and the huge promise of AI in healthcare in this field.
Great. Thank you. Yeah, no, I echo your points about it's a hugely evolving field and exciting developments every day. I don't have a slide per se, but I'll just comment that I think, as I mentioned earlier, areas in machine learning that can use minimum amounts of data. For example, we've seen developments in one-shot learning. And I think that's going to be extremely important for the later phases of drug discovery. In addition, as John had mentioned, unsupervised learning is also a very interesting field, tying back patient data all the way back to the individual design of the small molecule drugs. And I think that's going to be a very interesting field as we move forward. Thank you.
Sorry. Now, continuing a bit of theme around the data, what types of data and metadata do you both use? So Jeff first.
Great, thank you. Well, it really depends on the task at hand, but we don't throw anything away. For example, a simple example, compound color could be related to purity. And in some cases, color might be reported, other times a numeric purity might be reported, and there could be ways of combining those two data, and you could not do so if you threw both away or threw one away. So as a general rule of thumb, unless we truly believe we don't need it, we generally do not throw anything away, and it's very task-centric. Over to you, John.
Yeah, thanks, Jeffy. We are also loath, to not use data because, as you mentioned at the very outset, that data is normally very expensive to have been obtained, right? So we want to use it as many times in ways as possible. So of course, we use all directly relevant data, curating it carefully to give ourselves the best chance of success. But also, as I mentioned with the work that, described by Hugo Colemans and others, really see what other data can we repurpose, okay, to be applied to the problem at hand. Also at Numerate, we've shown that multi-level AI-based predictions can be used as metadata of their own type. Okay, predictions about conformational landscapes of small molecules, those are predictions. Okay, predictions about small molecules activities across a broad panel of predictive models, those outputs, okay, those labels, predicted labels, can be used, as we've shown, as the features to the building of a higher level metal model, especially in the context of predicting phenotypic activities such as antibacterial activity. So I think of this approach really because I'm amazed that my colleagues have shown that this is possible. I'm really, really impressed by it. I consider it as transfer teaching rather than transfer learning because of the fact it really speaks to the underlying predictive accuracy of the models that use these predictions such that they can successfully be applied themselves as data.
Thank you very much. Another sort of question that seems to pick up on some of the themes that are coming through on the Q&A is how do you maintain a competitive advantage in model building? And maybe explain how software tools can help with this. Right. Jeff, first on that one.
Thank you. So staying on top of new developments in the field, I kind of harped on data, but also, new algorithms are paying attention to new algorithms or new ways of looking at data is also very important. And proactively finding new sources of data is key. And that's, one of the reasons why we had set up these academic collaborations, finding new sources of data. In terms of tools and organizing data cycles, in terms of model building on a regular schedule and record keeping on why or how things were done in terms of how to build models. I think tools to record keep and annotate code are very important. So all these, the support infrastructure around model building and data curation are also extremely important tools that we use. We can compare code later to what was done in the past, so we understand why it was done and how to do it. And history has a habit of repeating itself if you're not careful. So those are extremely useful. And ultimately, somebody has to apply the new models and to real world problems. So it's important to interface the model builders with the users of the models and the scientists. And as John had mentioned earlier, we do focus on building a multidisciplinary team that is able to not only understand the data, build new models, and ultimately use them for real world drug discovery problems. And I think it works very well when all these things are in sync. Thank you. Over to you, John.
Yeah, and here, as in any rapidly advancing field, it really is critical to commit to remaining at the cutting edge. Okay, so that's easy to say, but also to thinking more practically. As we think about how advances and potentially new types of systems, how they may be integrated with current best practices, in your organization, both in terms of your in silico efforts, right, your modeling efforts, your underlying computational technology, as well as your laboratory realization approach, right? Okay, again, finding this resonance between your computational technologies and AI systems and their interaction with the techniques you're actually going to use to demonstrate things in the laboratory. So yes, I think of tools. It's amazing to think of like an entire platform of AI models and silico assays, as tools. But right, if in fact they are updated on a regular schedule, as Jeff mentioned, they actually are tools, and that really differentiates, I think, the work of drug hunters today and in an organization centered around application of AI from that of the past and traditional methods. And so Jeff mentioned, having just access to tools for record keeping, et cetera, and having them, again, they have to be modified and integrated in a way that's different than, in other types of organizations or approaches to discovery. And, under the current, COVID impacted circumstances, I would add that we found that, we're finding the use of collaborative workspace tools, that typically come from the high-tech community. Okay, open workspaces, Slack, Atlassian products. Those are really allowing us to maintain connectivity and productivity, even though we're working from, hundreds of home offices, right? And that's turned out to be very useful under the present circumstances.
Thank you very much. Last question before we move on to Q&A, but for me, I think this is the most interesting one. So whose work do you respect in this sort of space, and which researcher or breakthroughs, techniques, software have had the sort of biggest impact on you? So Jeff first for this one.
Great, thank you. So I've had a lot of great mentors that I really have to thank over the years. Throughout my initially in my training in organic chemistry, I really have to thank Dr. Louis Berriot at the University of Ottawa and Paul Wender at Stanford. And then as I went into the professional drug discovery, Brad Morgan at Cytokinetics. And I also have to thank the tremendous coworkers I have currently at Atomwise. And they've taught me, Abe and Izzy have both taught me a lot about machine learning for drug discovery, and Kong, my close coworkers at Atomwise. So in terms of the overall field, what had really sparked my interest in the first place is the work on RF score by John Mitchell and Pedro Ballester. They really sparked my interest in terms of using machine learning in drug discovery in their initial work. I also was really influenced by the work of AJ Jain and Ann Cleaves, who I know quite well, and who have been very prolific contributors in the space for a very long time. I really respect their work as well. Thank you.
Yeah, and I similarly have been very fortunate in my academic training, with Mike Doyle as an undergraduate, and Peter Dervin as a graduate student, and Chris Walsh as a postdoctoral. And then actually was kind of able to do a second PhD when I, left Stanford to become CSO at Theravance, where in fact we brought in a lot of, industry experience and very successful people, often from Merck, there to build that company up. I mean, our chairman was Ray Vagelos. Our first head of research was Bert Christensen, and I really did try to learn as much from them as I could. In the area of AI and applications of machine learning to drug discovery, I really do want to acknowledge my numerate colleagues with whom I've had the chance to work with, not quite 2 decades, but since 2003 in some cases. So really together, we innovated and persisted over the intervening period, which wasn't always, really very receptive to new ideas coming from the computational community, because we knew there was a there, not just in the computer. We had shown that things worked in lab and we could make prospective predictions that really made a big difference in how we practice drug design. So it turns out that we weren't wrong, okay, but we sure were early, okay, starting in that period. And now we're very pleased that we did persist to the present time, so that, we can help, along with our integral colleagues, to help realize the potential for AI to help patients.
Wow, very good, guys. John, I'm going to just open it to questions. We do have some time, so Charlie will read some questions for the panelists. You could, Charlie.
Yeah, thanks, Frank. Thanks, everyone. And John Overington, I know you've been monitoring the question panel as well, so if there are any favorites in there that you see, there's no way we'll get to all of them. There were quite a few good questions in there, so attendees, please forgive us for not going through every single one. It would be a completely separate probably hour-long webinar to get through these questions. I was interested in this one, though, for the panelists. Is there an example that you can share or maybe talk about of a surprise finding or surprise result that was unexpected that you found using machine learning or AI techniques? Is there anything standing out in your minds surprising?
I guess yeah, I would say that it's not so much surprising, but it's gratifying in the sense to find the general applicability of some of these techniques. And that's what makes, I think, so many of us in this field so excited that, in fact, okay, we can build a predictive model using this kind of representation. Say, for example, 4-point pharmacophores that were originally pioneered by Jonathan Mason to really capture the way molecules can present functionality in 3 dimensions over time, and that could be generally used as a way to capture really that phenomenon in a way that could also be made relatively scaffold independent, and that would then allow you to hop from scaffold to scaffold. So that's so very different than previous techniques based on consensus modeling and things. It really did represent a really quantum leap forward, and that's actually what led me to get involved with my numerous colleagues in this field as a whole.
Yeah. Thank you. Yeah, I have similar parallel stories. As I had mentioned earlier, our technology is a structure-based approach, but we find a lot of people do come to us with no structure is available, and we've actually seen successes in those cases where we've had to build one based on homology modeling, where there's no previous structure available, and we've definitely seen successes there. So I found that particularly surprising, given that there is no structure known. There's probably, in those cases, there's no known small molecules inhibitors of the targets, and yet we are still able to find success in those cases. And I find those cases very surprising still. Yeah,
and following up on that, Jeff, I would say that just this concept of homology and the fact that we can repurpose data along the lines we've discussed, right? That what it shows is there are some underlying core principles of how small molecules interact with proteins, for example, that can be transferred to many different situations. And that's, again, both surprising and gratifying, just to help us solve problems.
Yeah.
If I could maybe leap in with a question from the panel. There were quite a few sort of questions around sort of career advice, so the best training, the best sort of trajectory to get into this field and so forth. So maybe you could both comment on that.
I still find a lot of value in lab skills because I find those are more difficult to learn after professional training. But I think, also there's a lot of online tools to learn in silico programming languages, and you have to really do both, I find, and I think there's maybe a bit biased, but I still think there's a lot of value to understanding the principles of small molecules organic synthesis, and that's very, very important especially if you're in the field of small molecule drug discovery, but also being cognizant that there's a lot of in silico tools that we can use to better predict about where to invest our time, because as I mentioned earlier, chemical space is vast, and you can certainly spend an inordinate amount of time in the wrong area of chemical space.
Yeah, there's these parallels, right, between the number of molecules that are synthetically accessible, the number of stars in the galaxy and the universe. That kind of brings it into focus. And so, John, to answering that question, to me, I would really encourage people to continue to follow their passions and to follow them deeply, to become expert, right? I mean, a generalist who knows a little bit about organic chemistry and a little bit about bioinformatics and a little bit about machine learning is not the same as having a kitchen table around sitting people who are deeply expert in those areas and finding ways to communicate around the API translation issues to apply the really cutting edge and deep experience knowledge. So you continue to follow your passions, but also to really work at interfaces here, which is where a lot of the rapid advances are being made. And those are the interfaces where I think they have the greatest chances to benefit society.
Perfect. Yeah, thanks. Yeah, there was definitely questions on, you know, are there fellowships and training programs? So I love that. It's a good future for everyone. A quick question for the panelists: is the AI techniques really mature enough for use in a biologics design and discovery platform where the data apparently is even more sparse? I know you've touched a bit on you know the data that's needed or required or preferred, and even when you have structures versus not having structural information. What are your thoughts on that and the use of AI within a biologics design and discovery organization?
Yeah, the questions and the issues are different, and so, you know, for example, I would actually, you know, spoke recently to people at a small company called Peptone, who are, you know, informatics and machine learning people who are worried about this, primarily from the perspective is that, hey, you know, unlike small molecule drug discovery, where we may be even AI enabled, you know, designing and synthesizing and testing hundreds of compounds, if not thousands or tens of thousands in more traditional approaches. And then we start to worry about the, you know, the properties, CMC properties related to manufacture as we get closer to the end of optimization. In biologics, it really pays to worry about these things early on, because if you have a protein that aggregates or is insoluble, you know, you're not making millions of them, you know, it serially. So you need to worry about that early, and they're developing techniques that go beyond, I think, the very useful Rosetta toolbox for understanding and predicting properties of proteins, to deal with the edge cases and more challenging cases. So there, yes, AI is being applied in that field as well. The representations and the approaches are necessarily different, but there really shouldn't intrinsically be you know, reasons why, you know, properly approached these kinds of techniques can't make big difference there as well.
Yeah, absolutely. And I'll just echo everything John said. I think there's also a lot more opportunity in that space for rapidly evolving the data as well. You know, and developing the algorithms by generating new data. So I think that field is certainly going to see advances and will mature into the future as well.
Yeah, and you know, and again, it's really interesting to see, you know, the Vijay Pandey's, you know, folding at home effort, the David Baker's gamification of protein folding problem. These are really amazing efforts, and I think very productive efforts that are going on in that realm.
Perfect. All right, guys, to be respectful of time, I think we'll end up with one more question here. Kind of relates into the data. We've had some great discussions around the data involved or required in AI, and some great questions about that. Someone has asked, one of the attendees here, how willing or reluctant are the big pharma companies to sharing their full data sets in the early drug discovery phase, especially data on molecules who may have failed, because that, of course, would be a more abundant data source, and also the data on the molecules that are no longer interesting for the company. But as you pointed out regarding negative data, it should still be very interesting for learning and training purposes. Are you finding they're open to this sharing or making that data available in these efforts?
It's yes and no. Go ahead. Go ahead.
Yeah.
Go ahead.
Yeah.
A really quick sort of response from me on that. So in the European Union, I've got one of these innovative medicines initiative projects where I guess the technique of federated learning is being used. So the sort of safe sharing of data across a set of participants. Everyone benefits. You don't need to disclose your underlying proprietary data. So that's a project called Melody, spelled I think with 2 L's. So, you know, people may want to look at that, but there's plenty of other examples where there is engagement. I'll pass on to Jeff and John for that.
Yeah, I think in literature, there's certainly been cross-company collaborations around ADME data, where the data could be shared in a way where the structures are obfuscated and the descriptors were shared. I'm well aware of a paper recently in the field about that. And yeah, there are probably ways of sharing sensitive proprietary data, which still enables the use by the community, but still protects the private company's interests.
Yes, and again, you know, there's a firewall around just who's able to federate, right? So it's not open data like the Kemble data set, but one has to be, I think, sympathetic to the requests you can imagine scientists making of corporate lawyers that say, hey, we're going to share our proprietary structures and the proprietary data that goes along with them. So I'm hoping that, and my belief is that over time, pre-competitive data, post-competitive data will truly be recognized as such, and that it will be shared more broadly. If you think about, for example, what needs to go into patents, really you're looking for a molecular structure that has a novel molecular structure that has a specific utility. Typically the utility is an on-target activity or a reflection of that on-target activity. All the other properties, you know, related to physicochemical properties, membrane penetration, ADME-related properties, those things are, you know, those are not, you know, necessarily, depending on the kind of patent you're writing, are not really key to that. So perhaps there will be opportunities to share different kinds of data or different aspects of data in more open settings, especially as Jeff alluded to earlier. That kind of PK ADME and toxicity data is the most sparse of all, and it's typically also the most noisy from my experience.
Yep.
You guys know we're running out of time, so I want to thank John Overington, John Griffin, and Jeff Warrington for a great discussion today. It was, I think we could probably go another hour without fail, considering the questions and just the topic. So you guys, thank you again. So to finish out, of course, I have to tell a little bit of a story. So for those of you interested in hearing more about CD Vault or even our newest offering, BioHarmony, drug data store or annotator, the place to find out more is to come to CD Vault. If you're looking for CD Vault, come to collaborativedrug.com or email us at info@collaborativedrug, and we'll be happy to work with you. If you're interested in hearing more about what's coming with BioHarmony, reach out via email to info@biometadata.com. One more thing. So upcoming events, what's coming next? So in the course of the next several weeks, we'll be at Discovery on Target, which is actually next week. We'll be exhibiting and we'll be presenting CD Vault in a demo and a talk. And also we'll be having a joint symposium with Optibrium with their StarDrop tool and collaborative drug discovery in October, where we'll feature both StarDrop and CD Vault working together, as well as a November event. We'll be working with Certara when they feature D360 along with collaborative drug discovery, CD Vault, in a webinar sponsored through BioIT World. So again, any other questions, feel free to look at all the details down below in our blog. If you click on that link, or actually copy that link, you'll find a place to find all about our webinars and events and all kinds of great news from Collaborative Drug. So gentlemen, again, thank you so much. And we really loved working with you guys on this event. So we hope that all of you will join us for future events with CTD. So thanks again for your attendance, your questions, and your participation. Thanks again. Have a great day, everyone.
Goodbye.
Bye-bye.
Bye. Thank you.
Bye.